As you said, I'm working on bees and honeybees from 2018. And today I'm going to make a presentation about bee venom. So I guess there's a problem with the... Yes. So when we are talking about the bees and venom collecting on bees, we are working on honeybees. There are too much species that we call bees, but we are working on Apis mellifera. And they, as you know, they are social insects and they live as a colony. That's why we are. It makes us easier to collect their venom. So there are 3 individuals, 1 queen in each colony, hundreds of drones, males, and thousands of female workers. And their life cycle, they all have same stages to egg, larva, prepupa, and pupa, and they become adults in 3 or 4 weeks, depending if they are queens, workers, or drones. And if you want to talk about bee venom, we have to talk about bee stinger. So bee stinger is the most important defense and attack organ of the bees, especially in honeybees. And in honeybees, bee stinger is a structurally modified form of the ovipositor. That's why only the queen and the workers have stings, and males doesn't have venom sacs or stings, and they cannot sting. As you see in the picture, the queen stings and the worker stings are different from each other. Queens always use their sting to defend with the other queens, and workers are the guardians of the colony. And the bee venom is secreted from venom sac, located in the last segment of the worker bees and queen bee's abdomen, and it's about 2 millimeters long and has a pointed tip, and the venom is stored in the venom sac. And the stinger of the bees physically consists 2 parts, venom sac and the needle. And you know, when honeybees sting, they lose their life because they lose all their stinging apparatus, including the muscles, nerve center, everything, and they are always fatal for the bees. When this stinging happening, they are just giving a alarm pheromone, and this pheromone is like a warning, a sign to the other bees to attack the same location. And we know that the amount of venom stored in the venom sac is approximately 0.3 milligrams, but in young queen bees, that is about 0.7 milligrams, but we are not getting bee venom from the queens because, you know, queens are everything from the colony and we cannot harm them. And bee venom production reaches the highest level in the bees that are 16 to 19 days old. The ones that we call the guardian bees, they are always in front of the hive and guarding the hives. So when we look at the physical properties, bee venom is a liquid, light yellow, odorless, and it has a sharp and bitter taste. But we are collecting the dried form of the bee venom. I will show you how to collect it. And bee venom is soluble in water and consists 80% of water. And the main part of the honeybee venom is peptids, proteins, phospholipids, biogenic amines, sugars, volatiles, and minerals. And if we are talking about apitherapeutic or cosmetic uses, melittin is the active ingredient, and then phospholipase A2, MC peptide, apamine, and histamine. Especially melittin is special for the honeybee venom. So the amount of and the characteristic of the venom changes by different geographical regions, different honeybee races or ecotypes, different collecting methods, altitude, weather conditions, pollen and nectar sources, queen bee age, colony population level, feeding and collecting periods. So if you want to get honeybee venom, we have to be careful about all these criteria. So there are two ways to collect bee venom. One of them is directly collecting from bee, individual bee venom, but it takes long and you can get less venom from one bee to other one. The process that we use is electroshock method. So it's a method that bees which are placed on a copper wire and there's a glass surface under it, and when bee comes to the wires with the electricity, they pour their venom onto the glass surface. And here you can see how to collect from the landing board in front of the hive and top of the hive. You can collect in each methods too. And we are using this method because I told that they are losing their life, but with this method, none of the bees lose their life. So we can get bee venom directly. And after drying, we are waiting for 2 or 4 hours in a well-ventilated room, and then just scraping the venom, dried venom, from the glass surfaces. We used black surfaces to make the aggressive on the bees. So where can we use bee venom? Bee venom is using in pharmaceutical industry, cosmetics, and apitherapy. Bee venom is actually using from ancient times to Romans and Egyptians as a treatment to some diseases, and you can use bee venom both in injection, creams, or tablets, and etc. So the use of bee products in human health called apitherapy. So in apitherapy, we can see both ways, bee venom therapy or bee sting therapy. And we know that bee venom has too much effects on human health, like increased blood circulation, bactericides, regulator of heart rhythm and functions, protection against radiation, blood pressure lowering effects, cancer therapy, and too much other effects. And some studies on human health, you can see the effects on cancer cells, liver fibrosis. Also, we can use bee venom studies on animal health too, because we are working on agriculture and on farm animals. So bee venom therapy could be an alternative for the broilers, for the dairy cows and their diseases. So studies show that bee venom has a strong performance against some critical diseases. Therefore, more scientific studies are needed to determine appropriate standardization and appropriate dose usage. Actually, bee venom production is newly developing in our country, because our country has a important place for beekeeping, but bee products are not producing too much for now. And the toxicity of bee venom, it's an important point, because bee venom allergy could be so serious, and the median lethal dose of bee venom varies between 2.8 and 3.5 milligram of venom per kilo of human body. It can thus be speculated that a non-allergic person weighing 60 to 70 kilos has a 50 chance of death upon being stung by 1,000 and 1,500 bees. And these are some pictures that we observed in the field. In the right side, you can see a snake bite venom, and we show the similar allergic reactions in our bodies. So at last, I want to talk about our project. We are together working with Mr. Susmut, Hemfel, and Maik together in this project. So determination of chemical content and bioactivity of Anatolian bee venom collected from different geographical regions of Turkey. So we have a huge field trip for this project, because we know that bee venom composition and the amount can change by region and races, and Turkey is a big country, and we have, yes, main 5 bee species, but way more ecotypes in our country. That's why we wanted to make a huge field study and take all the samples from the locations. So the aim of the project was to characterize the chemical content and bioactivity of honeybee venom by making regional samples. And this study is the first detailed study to be conducted to determine the venom quality of honeybee species in Turkey. With this project, we will be able to investigate and compare the honeybee biodiversity of Turkey by applying geometric morphometric methods. And within the products, it's expected to discover a new molecule, maybe a new structure of melittin or other new peptides. And we believe that this study will be a leader project for regional project. So this was our working plan. And these are our locations. It was 53 apiaries in all over Turkey, and we were in the field for 3 months, maybe 15,000 kilometers. It was a huge trip. Yeah, so it's all about the geographical differences, plant diversity, and biodiversity of honeybee species in our country. So these are my references, and thank you.
Thank you, Ekin, for the talk. Are there any questions from the other people?
Yes, I have a question. When you are drying the venom, are there any components that are easily broken down or like that are degenerating easily?
Jacob, is actually we lyophilizate our samples as soon as possible, because we know that these things can happen. And in the bee venom, we can see the different by colors. You know, when you are just past the time, it just turns to a yellow brownish color, and you know, all the components are not that good. So we took all the samples lyophilized.
A question from my side, would it be possible to force the development of queens to investigate the venom of queens without damaging a hive, so that you take some larvae and then generate your queens on your own without any colony that would be then endangered?
It would be because you know, we are also producing queen bees in apiaries. You know, you can produce, you can make colonies to make their own queen, or you can create new queens and give to them. But when we are thinking about this project, we were always thinking about, okay, we have colonies, we have production, and we can produce bee venom from the colonies. But yes, it could be. It could be.